Hello and welcome everyone to Biology 572 Epigenetics. I'm really excited to be teaching this course for you on epigenetics, in part because it's such an exciting time to be studying epigenetics. In many ways, epigenetics is changing many of our views on genetics and biology as a whole. Some of the things I learned when I took genetics about 25 years ago have changed dramatically because of our understanding of epigenetics. Nessa Carey, who is a leading epigenetics researcher, summarizes where we're at today in epigenetics in this statement. Epigenetics can potentially revolutionize our understanding of the structure and behavior of life on Earth. Epigenetics as a discipline has been around for decades, but really within the past 10 to 20 years it has received a lot more attention. It's not surprising it could be changing the way we look at genetics as we know it. And remember that genetics itself as a discipline is only about 150 years old, so it's a relatively young science. If you remember back to when you took your undergraduate genetics course, in the early 1900s, when Mendel's works were rediscovered, scientists were trying to understand how these traits could be inherited. And then they spent several decades doing experiments documenting and understanding the inheritance of this genetic material. Then in the, say, 30s, 40s, and 50s, there was a push towards understanding what the genetic material was and then the structure of the genetic materials. If you remember some of the experiments from your undergraduate genetics course, such as the Avery, McLeod, and McCarty experiments, the Hershey-Chase experiments, Rosalind Franklin's work on the structure of DNA, and Watson and Crick's work on it as well. Once they discovered that DNA was the genetic material, and they discovered the double helix was the structure of DNA, they really felt that they had the key to life in their hands. And over the next several decades, researchers worked on trying to understand how DNA was copied, how it coded for traits, how it could be inherited from one cell to the next cell, or from a parent to a child, how it could be mutated, and how those mutations can lead to different traits or even diseases. They studied how it was regulated so that different cells which had the same DNA could make different proteins. They studied how variations generated through meiosis could lead to the diversity that we see on Earth. In fact, understanding the structure of DNA and its inheritance really helped us understand evolution in much finer detail. Then if you fast forward all the way up till when I was in graduate school, say in the late 1990s and the early 2000s, there was a big push towards understanding the complexity of genomes, the sequence of the entire genome of particular organisms, understanding the transcriptome and the proteome, that is, understanding which messenger RNAs and proteins were being expressed in which cells in a tissue-dependent or stress-dependent manner. Understanding all these sequences and how they interacted. At that point, we thought, now we truly could understand the complexity of life. And it did go a long way. Now, as science often does, once we think we understand something, additional complexity enters the picture. And this is exactly what epigenetics is doing. It's a relatively new field, as I said earlier, but this relatively new field is making us rethink much of what we know about genetics and biology in general. So I'm going to move over to my whiteboard to introduce you to the field of epigenetics, but also to the course. Okay, so let's move to that whiteboard. Okay, what I'd like to begin with here is a definition of the word epigenetics. So this definition I just pulled from the internet, but it's a pretty good one. The study of changes in organisms caused by modifications of gene expression rather than alteration of the genetic code itself. Okay, in addition to this general definition, I'd also like you to know these other features of the definition of epigenetics. First, you should know that epigenetic traits are heritable. And when I say heritable, I mean that they are inherited from cell to cell during cell division and from parent to child in a generational effect. The next thing is that these epigenetic changes are reversible. They're not necessarily readily reversible. Often it takes a great deal of energy to make changes in epigenetic expressions, but they can be changed. And the last one is one of the most important features of epigenetics, and it's kind of implied in this definition, but let's put it in words here. Epigenetics is not a mutation. The effects we see are strictly due to the change in expression and not to a change in the genetic code itself. In thinking about epigenetics, let's begin with a fairly generic view of two genes. These are the exact same genes, and we'll just call them YFG for your favorite gene. The sequences are the same. There's no difference in them. They have the same promoters, and again, the sequences are identical. It's possible these could be in different people. It's possible that they could be in the same person, but different tissues. Could be in different stages of development. Maybe in one situation, a particular stress is applied to one gene and not the other, or one cell containing this gene but not the other cell. But the end result in this example is that this top gene, let's say, is expressed, and this bottom gene is not expressed. Throughout the course, when I use an arrow in this manner, I mean the gene is activated. It's turned on. When I use a bar, such as shown here, I will intend that to mean that the gene is not expressed, that it is turned off. And the reason one is on and one is off has to do with the accessibility of regulatory elements. It could be access to the promoter, it could be enhancer or silencer sequences, other upstream regulatory elements, but ultimately, whether or not transcription factors can reach these elements is of prime importance in understanding epigenetics. Often, when we think about epigenetics, we think about environmental factors that can control this, can control whether or not these regulatory elements are accessible. And so what are some of these environmental factors? There are too many factors that we could list here, but I'm just going to give you a sample of some of these factors that can regulate whether or not these genes are on or off. This first, let's say, is nutrition. Various nutritional factors can regulate these expression of these genes and can have an effect on the regulation of certain genes for many generations, in fact. Various kinds of behaviors. These could include tobacco, alcohol, and other drugs, both legal and illegal. We know things like the behavior of parental care can have an effect on gene expression. The amount of sleep one gets can have an effect on epigenetic expression. And many other behaviors, some of which we'll talk about throughout the semester. Various kinds of stress. These could be chemical stresses, these could be psychological stresses. Various kinds of stresses can impact the expression of genes. Physical activity, the amount of exercise you get. Pollution, we know, can affect epigenetic expressions of genes. Now, when we think about all of these environmental factors I've just listed here, it should be clear that this is at any stage of development. This could be factors that affect a baby as it's developing in utero. It could also be factors that are affecting a child or an adult. I should also remind you that when we talked about epigenetics, we said that it could have a generational effect. So some of these behaviors and effects one individual experiences in their life can change that expression of those genes for their kids, for their grandkids, their great-grandkids. Now, we'll certainly talk a little bit about these environmental factors and how they influence epigenetic expression. But what are the molecular mechanisms that these factors trigger to change the expression? It's one thing to say that nutrition has an effect on gene expression, but how does nutrition have an effect on expression? And that's where we're going to spend a lot of our time this semester. So let's just give you a brief intro into these molecular mechanisms. Some of the big ones that we'll talk about include DNA methylation. And when we think of DNA methylation, we're primarily thinking about the promoter. So we'll talk about how that happens, the enzymes that influence whether or not a promoter is methylated, what happens when it's methylated, and all that good and crazy stuff. The next includes histone modifications. In your genetics class, we talked about acetylation and deacetylation, but what we're going to learn in this semester is it's more than just acetylation and deacetylation. There are many other kinds of ways that histones can be modified. And then we'll talk about the effects of those modifications and the enzymes that are important in modifying the histones. The last thing I want to mention right now is the expression of regulatory RNAs. These include microRNAs, and they also include long RNAs, like we might see with X inactivation. It's also important to bring in the evolutionary aspects of epigenetics. We'll certainly go into a little bit more detail on this concept later on in the semester or throughout the semester. But for now, I think it's important for us to know that we see evidence of epigenetics in all the domains of life. In bacteria, now what we see in bacteria, it's not going to be quite as extensive as what we will see in eukaryotes, but it's still there. We also see examples of epigenetics in archaea. Turns out what we see in archaea, it's not as well defined because it hasn't received a lot of research attention, but what has been done shows that there are epigenetic effects in archaea. And in some ways, in fact, many ways, it'll be more similar to eukaryotes than bacteria. One way, for instance, archaea has histone-like proteins that are more similar to gene expression in eukaryotes than bacteria. And if you remember from Biology 111, archaea are more closely related, evolutionarily speaking, to eukaryotes than they are to bacteria. So it's not surprising that they're going to use similar mechanisms. And remember, in eukaryotes, we have these 4 kingdoms. So animals, we'll spend quite a bit of time with animals. We'll spend some time with plants, particularly when we talk about genomic imprinting and how that impacts angiosperms but not gymnosperms, fungi, and protista. We probably won't say anything about protista after this slide, but we do see some evidence of epigenetics in protista. Now, to gain a greater appreciation for why epigenetics is important, I think it's important for us to talk about who studies epigenetics. Clearly, biologists study epigenetics, but more specifically, geneticists and molecular biologists study epigenetics. In addition, evolutionary biologists are interested in epigenetics. Ecologists and conservation biologists study the effects of epigenetics. In fact, it's very important in understanding these fields. Behavioral biologists are very interested in epigenetics. Neuroscientists are interested in epigenetics. And the last one I'm going to list here, though I could make this list a lot longer, and those are biologists who study disease. Epigenetics has greatly helped us understand the nature of diseases and how they're inherited and what causes them. And so maybe we could also put epidemiology here as well. We also can make a list of fields that are not necessarily biologists, but they're also interested in epigenetics. The first few I'm going to list here are important in understanding the socio-economical impacts of epigenetics. And that includes sociologists, social workers, and economists. In addition, psychologists also are interested in the epigenetic effects in psychology. Nutritionists are interested in epigenetics. Those working in public health or global health are interested in this as well. And I've been reading a couple interesting articles on how historians are using epigenetics to explain various historical questions. The last one I want to put here are those in the medical field. Physicians, PAs, nurses, dentists, pharmacists, and many others are interested in epigenetics so that they can best treat their patients. Now, I could add many more examples to each of these lists, but the main take-home point is that epigenetics has a broad impact upon many scientists and many professionals. We'll spend most of our time this semester here, but where appropriate, we'll bring in examples below here and over here on the right. And as you begin to think about your mini-grant proposal, you may want to explore some of these other areas on your own to pick a very competitive proposal. All right, this is all I want to say about the introduction of epigenetics. I do want to spend the last few minutes here talking about the organization of the course. So as I'm thinking about this course, there are three main areas that we're going to learn about. The first is understanding some basic concepts from genetics. This we'll only spend about a week or so on. We're not going to redo all of genetics, but I do need to remind you of some relevant concepts from genetics. These would include Mendelian or transmission inheritance. Even though epigenetics changes the way genes are expressed, inheritance remains the same. And I definitely need to spend some time talking about transcription. Epigenetics is all about transcription, so we need to understand that, and so I want to spend a little bit of time reviewing transcription with you. Linked with transcription, we need to review some of those molecular structures in a cell, particularly things like chromatin and its impact on gene expression. We're going to talk about how epigenetics can affect phenotypes. We'll spend quite a bit of time on this middle section here, and we're going to focus heavily on molecular aspects. And lastly, we'll talk about epigenetic effects on disease, including cancer, and other traits. These will be the three main features of the course. Now, as we're going through these, and where it's appropriate, we'll talk about examples found in animals, in plants, fungi, and probably a couple examples of bacteria as well. So now let's talk about my approach to learning. I use an approach called team-based learning. And sometimes I just call this TBL. I've used this approach all the way from 100 level courses, both for non-majors and majors, all the way through upper level biology major courses, 500 level, 600 level graduate level courses, and I've also used it in teaching medical school classes. In all of these cases, what I see is an improvement in overall grades, particularly exam grades, and most importantly, what I see is an improvement in retention. Now, what I want to do is just give you a brief introduction to what TBL is, but if you've had me for cell biology, biology 112, or genetics, biology 211, it's the same approach. But in talking about TBL, it's important to differentiate what we do in class to what students do outside of class. What you do outside of class, that's the stage that we call preparation. Getting ready for that next day's class. And I have posted on Blackboard what I call note-taking guides. They're also essentially your study guides. And if you understand everything in the note-taking guides, you should do well on the next stage, which I'll talk about in a moment. But to help you with the note-taking guides, you have your textbook. I also have video podcasts that go over many of the main points. In the past, students have sometimes said they only use the textbook. Other students say they use a combination of the textbook and the video podcast to understand the note-taking guide. Now, ultimately, how you prepare for class is totally up to you. You can ignore all of these tools and just say, read the chapters for that day. These are just some tools I'm providing for you to help it become more clear what I expect of you for that next day's class. Then in class, students will take what we call an IRAT, which stands for Individual Readiness Assessment Test. Did you come to class prepared? Then in your teams, you'll take a TRAT, which stands for Team Readiness Assessment Test. These here are what we might call lower learning questions. And what I mean by that, these are not the applied kinds of questions. They might be more recall types of questions. Then after we complete these quizzes, and I should also mention that these two quizzes are the exact same. One you just take individually and one you take as a team. And then the third thing here is the application phase. This is where you apply some of that knowledge you learned before class and we assessed you on with these quizzes. And we'll work on various problem sets together. We'll work on case studies together to bring some real application to what you're learning. And then what you do the next day or before the next class is you start the whole process over again with preparation for the next day. Okay, that's all I have for this first podcast for epigenetics. If you have any questions at all, please let me know. If not, I will see you in class. Bye for now.